Adrenaline acts as a ligand and binds to a G protein coupled receptor. This causes shape change in the receptor. The receptor then binds in an active G protein, causing the GDP to change into GTP, which activates the G protein. The activated G protein dissociates from the receptor and binds to adenylyl cyclase, causing a shape change, which catalyzes synthesis of CAMP from ATP. The CAMP acts as a second messenger and activates the protein kinase A. The protein changes shape and activates transcription factors and sends it to the nucleus. This produces proteins that inhibit movement of food in intestines during flight or flight response. ETX is a toxin and it will replace adrenaline and binds to GPCR as a ligand. This causes shape change in the receptor. The receptor then binds in an inactive G protein, causing the GDP to change into GDP, which activates the G protein. The activated G protein dissociates from the receptor, then binds to adenylyl cyclase, causing a shape change, which then catalyzes synthesis of CAMP from ATP. However, CDX causes this process to happen rapidly, results in high levels of CAMP release. The abundant amount of CAMP then binds and causes a shape change in the chlorine ions channel. The closed ion channels will undergo a shape change, then opens up and the water/ions will come into the intestines, causing vomiting and diarrhea.
Transduction pathway. Insulin acts as a ligand and bonds to the inactive tyrosine kinase protein receptors. The two receptors are then attracted to each other and form a dimer through the process of dimerization. This process activates the tyrosine kinase regions of each polypeptide, causing a response of phosphorylation to happen, changing ATP to ADP, adding phosphate to a tyrosine on the tail of the other polypeptide. The phosphorylated dimer then serves as a protein receptor and activates IRS, which activates other kinases. This series of molecules that are activated causes the glucose transporter channels to be transported to the surface of the cell, which is opened and then allows glucose to enter the cell. In this pathway, the overproduction of insulin in the pancreas causes the kinase receptors to change shape. This means that the ligands are no longer able to bind to the receptors, inhibiting the process of dimerization and therefore inhibiting phosphorylation. This prevents the activation of the GLUT4 channels to move to the surface of the cell, so that glucose is not able to enter the cell.
External growth factor return to G1 binds to the tyrosine kinase receptor, activate the dimerization reaction. Dimerization then activates the tyrosine kinase region of each polypeptide. Each tyrosine kinase adds a phosphate from an ATP molecule to a tyrosine on the tail of the other polypeptide. RAS binds to the activated receptor, change shape, and get activated. RAS binds to adenylyl cyclase, causing a shape change which catalyzes the synthesis of CAMP from ATP. The GTP then get hydrolyzed into GDP and the RAS get disconnected from adenylyl cyclase. The CAMP acts as a second messenger, activates a series of kinase enzymes cascade, then activates transcription factors to the nucleus, initiate the transcription of mRNA for cyclins, then translation soon follow. External growth factors return to G1, binds to tyrosine kinase receptors, activate the dimerization reaction. Dimerization then activates the tyrosine kinase region of each polypeptide. Each tyrosine kinase adds a phosphate from an ATP molecule to a tyrosine on the tail of the other polypeptide. Hyper-RAS binds to activated receptor, change shape, and get activated. Deactivated hyper-RAS then binds to the adenine cyclase, causing a shape change which catalyzes synthesis of CAMP from ATP. The CAMP acts as the second messenger, activates a series of kinase enzymes. Cascades then activates transcription factors to the nucleus. This initiates the transcription of mRNA for cyclins. Gestation soon follow. Hyper-RAS is a mutated form of RAS, so it is unable to hydrolyze GTP to GDP, get rid of a phosphate group. This causes the hyper-RAS to stay fixed in the active form and keep producing CAMP. Hence, cyclins would continuously be made and the cells would skip G1 and divide rapidly, causing tumor cell growth.